Hello everyone. So in last lecture we have seen pharmaceutical aids part 1. In that presentation I have given you the basic introduction of pharmaceutical aids and we have seen the two drugs, right? So today we are going to study all the remaining drugs of the pharmaceutical aids category. So just let's start with the pharmaceutical aids. So yes, right. So you have seen the topic that is pharmaceutical aids and you have this topic in pharmacognosy. The flow of presentation, is that is introduction, then classification, example of drugs, then uses, right? Then the list of drugs you have in as per your syllabus, that is kaolin, lanolin, beeswax, acacia, tragacanth, alginate, agar, guargum and gelatin. Why I am revising this? So you can remember the drugs also. So if anybody asks you in the MCQs or multiple choice question, in objective question, definitely you can answer that question, right? So we'll move forward. Introduction. So you have seen the definition. AIDS means the substance which is having no therapeutic value or little therapeutic value, but which is used in the manufacturing and compounding of the various pharmaceuticals, which is called as a pharmaceutical aids. Okay, sources: animals, vegetables, minerals and synthetic, that we have seen earlier. Now we'll start with the very important tragacanth, the important drug of this category. The synonym of tragacanth is gum tragacanth. Biological source: it is the dried gummy exudation obtained from making incision on the stem and branches of astragalus gummifera. So biological source is, that is a dried gummy. Gummy means like a viscous solution. Okay, viscous exudation. Exudation means we can scrap from the plant. And the biological source is astragalus gummifera, which is belonging to the family Leguminosae. So important part in this drug, astragalus gummifera family Leguminosae. The geographical distribution, it is mostly indigenous. Indigenous means it is originated from that place. Okay. So वो उस स्टेट से या उस कंट्री से ओरिजिनेट हुआ है, like Iran, Greece, Turkey, Iraq and Syria. Then description, here you can see the image as well, this is the, we eat in our ladus and all, that is a gum, gum. So tragacanth is similar. It is having the pale yellowish white color. It is odorless. It doesn't have any smell or odor. The taste is tasteless. Obviously if you will mix with something, then it give you the taste, but basically it is tasteless. The flakes are 25 by 12 by 2 mm size of this when you will exudate from the plant. From this shape, it is a ribbon like flakes and less curved flakes, ribbon like shape. So it is having the ribbon like shape. Then the most important part, that is a chemical constituent. So if you want, you can take the screenshot of this and you can write in the similar way in examination. Tragacanth, which is having water soluble and water insoluble chemical constituent. Here in tragacanth, that is a tragacanthine, which is 8 to 10% present in the chemical constituent. Water insoluble, that is a basurin. So remember the two names, tragacanthine and basurin. That is the most important chemical constituent of tragacanth. Then you can see here the product of hydrolysis of tragacanth are galactouronic acid, D-galactopyranose, L-arabinoramnose and D-xylopyranose. Means many carbohydrate and sugars are present. The identification test here, so you can see when warm with the sodium hydroxide solution, it gives you the canary yellow. So that's why I have highlighted with the canary yellow color. So immediately it will click in your mind. Yes, canary yellow color obtained when the tragacanth solution take in test tube and react with the sodium hydroxide. Second, when you will react with the iodine solution, it gives you the green color. So here also I have highlighted that part so you can understand easily. The uses of the tragacanth, as a demulcent. So demulcent means it will reduce the irritation and inflammation on the skin. It is also used as an emollient in cosmetics. Emollient का meaning होता है it will soothe your skin. Okay, so it will give you the smoothness on your skin if you will apply. Confectionery, sweets and chocolate considered collectively. So tragacanth also used in many confectionaries. Maybe you didn't know the name, so I have added the meaning of this confectionery which is used in the sweets and chocolates. Mostly. Then next is it is used as a suspending agent, emulsifying agent, as a binding agent, and it is also used in the many cotton fabrics and in the calico printing. So those are the uses of tragacanth. Next drug is agar. Agar is, you have heard the name of agar in many practicals, in many things, right? Agar, and you know this is like algae. So synonym of agar is agar agar. Here you can see I have put the one diagram, that is, or image, that is agar agar powder. Biological source of agar is, it is the dried gelatinous substance which is obtained from Gelidinium amnesi. So remember the name Gelidinium amnesi. And there are many species of red algae. If you want to watch the video how to produce the agar, there is one video on YouTube, you can watch it. I will link that video in description. Okay. Family Rhodophyceae. The family of agar is Rhodophyceae. Next is geographical distribution. Agar mostly indigenous to, or it is produced commercially in Japan, Australia, New Zealand, USA and India. It is also in India, it is mostly produced in Bay of Bengal. You know Bay of Bengal. So I have put here the whole complete procedure of the preparation of agar, how from the seaweed, you know, seaweeds, or from bamboo, how algae is produced. So you can read this in detail. Like in Japan, the red algae is grown on the bamboo, spread in the oceans. Then collection of the material is done in May and October month. Then seaweeds are scrapped from the bamboos. That seaweeds will be scrapped from the bamboos, dried. Then you have to dry that. Then you have to shake that. So likewise, you will get this procedure how of the production of the agar. And then next is, you can read thoroughly and you can write it down, so you will get clearly. Next is description. In description here, you know, agar is color whitish and creamish color. Odor is odorless. Taste mucilaginous. Size, sheets are 45 to 60 centimeter long and 10 to 15 centimeter wide. At the end, the sheets will produce. So that sheets are 10 to 15 centimeter wide and long, 45 to 60 centimeter. Shape, you will find agar in strip shape, sheet shape, flakes, coarse powder. In many shape you will find this. Now the chemical constituent. Here I came for the important part, that is the chemical constituent. Agar consists of two different polysaccharides. You know the meaning of polysaccharide, that is the chain of sugars, that is agarose and agaropectin. So please remember these two drugs, agarose and agaropectin. And agarose is responsible for gel strength. You know, agar is having a gel strength. Gel means, you know, jellies like viscous and mucus. So agarose, because of this agarose polysaccharide, it is having a strength of like a gel strength. Okay, and this agarose is composed of D-galactose and 3,6-anhydrogalactose unit. And the last is agaropectin is responsible for the viscosity. So agar is very viscous solution and agaropectin is responsible for it. So these two main chemical constituents, agarose and agaropectin. Remember these guys. Then next is identification test. So it's very easy. You have to boil 1.5 gram of agar in 100 ml of water and you have to cool it. If it will create jelly, then it is pure. Okay, so same thing. Second test is similar like that. Then uses, here you can see the emulsifying agent. It is used as a laxative. It is used in the preparation of jellies, confectionery item. In the microbiology, you know, when you will go to like in second year, maybe you have microbiology subject. So in practicals, you know, agar you need agar for the culture of the preparation of culture of bacteria. So here the next use I have put the same, preparation of bacteriological culture medium. It is also used in the chromatographies. Next is next drug, lanolin. So far we have studied two drugs, tragacanth, agar. Now next is lanolin. Here you can see lanolin, also known as hydrous wool fat. Okay, it is you are getting from the it is a wool fat. So synonym is adapts lani. Synonym of lanolin is adapts lani. Biological source, hydrous wool fat is the purified fat like substance obtained from the wool of the sheep, Ovis aries. Please remember the name sheep Ovis aries belonging to family Bovidae. Then geographical distribution, that is Australia, USA and also in some extent in India. Description, it is whitish yellow, odor is faint and characteristics, and taste is bland. So here I have put the image, so you will 100% get it is used for the applying on the skin. This is a chemical test, same like the previous one. So I will directly jump to the uses, that is it is used in water absorbable ointment basis. So you can check on your Nivea cream, whatever cream you are applying on the skin, you will find this chemical constituent. It is mostly used in the cosmetic preparation. Next is the beeswax. So beeswax, yellow beeswax and sera flava. Synonym of beeswax is yellow beeswax and sera flava. Biological source, yellow beeswax is a purified wax obtained from honeycomb of the bees Apis dorsata family Apidae. So that is very important, the family is Apidae. Color, as you have seen, yellow to yellowish brown. Odor, agreeable, it is having a pleasant smell, that is agreeable, or we can say honey like smell. Chemical constituent, that is myrcene, myrcyl palmitate, and it is having a free steroic acid also. Uses, mostly it is used as a what we can say for the candle making, for the face cream, it is used in an ointment, it is used for the plaster and polishes. Then last, that is sodium alginate. This is very important. Sodium alginate, algene sodium polymeronate. Biological source, sodium alginate, algene sodium polymeroate. Okay, the name of this is algene sodium polymeroate. Biological source, sodium alginate is the sodium salt of alginic acid composed of a manuric acid and glucuronic acid. So please you can take the screenshot, so you will remember like how to write this and what is the alginic acid. Family Phaeophyceae. Family is Phaeophyceae. Geographical distribution, seaweeds are mostly found in Atlantic and Pacific Ocean, mostly in Japan, USA, Canada and Australia. Description, color is white to buff color powder, odorless, taste is tasteless. It is uses of sodium alginate, it is used in the paste, creams, thickening and stabilizing emulsion, good suspending and thickening agent, it is used in the jellies, it is used in textile industries. So those are the uses of alginate. Now next is gelatin. This is very famous and nowadays we are using gelatin sheets, gelatin paper, right? So, and you know this is produced from the animal tissues like skin, tendons, ligaments, bones. So, and in this diagram you can see how gelatin look like, right? So, colorless to pale yellow, odor, characteristic odor is there. Taste slightly like a broth like, because it's very viscous, like, and it is found in sheet, flakes, cores and fine powder. Chemical constituent are moisture is present, then gelatin, you know, mostly gelatin. It is used in the preparation of candles, jellies, thickeners, jams and etc. Last is very important, kaolin. The synonym is china clay, then porcelain clay. It is made up from the hydrated aluminum silicate and free from gritty particles. So please remember the biological source. So mostly in exam they will ask you the synonym and biological source of kaolin. There are three types: heavy kaolin, light kaolin and natural light kaolin. There is little difference of the dispersing agent. Description, it is a normally white or grey, yellow, brown color. Odor is odorless. And the size of kaolin is 10 micron to 690 micron, depending upon heavy kaolin, light kaolin or this natural kaolin. Uses, it is used as an absorbent in oral administration, it is used as a disinfectant powder, it is used in filter, paper, rubber and etc. So those are the links from where I have found, like I have made this PPT. Thank you for watching. And I hope you will like this explanation on the pharmaceutical aids. Please like my channel and subscribe to my channel. Again, thank you for watching. If you have any doubts, queries, please feel free to comment in comment box. Thank you very much. Thank you.